какой неправильный. Алло, 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 алло. Угу. Good morning. Today the lecture is Physiology of Bacteria.
Good morning.
Where are the students are? Inform
Don't
them
know.
they have a lecture now. Could you write your leader, please? And other
Okay.
students?
Okay.
The lecture is Physiology of Bacteria. Physiology of Bacteria, a section of microbiology in which study bacteria nutrition, the second energy metabolism, the third growth and reproduction, fourth enzymes, and fifth pigments. Bacterial nutrition. Bacteria are living cells, so they need nutrition. And in accordance with the type of nutrition of living organisms, autotrophs and heterotrophs are distinguished. So bacteria are divided on autotrophs and heterotrophs. What are they? Autotrophs. Autotrophs are living cells. Those are able to use carbon dioxide or carbonates as carbon source, and they are used in nature as usual. Saprophytes, a lot of microbes are saprophytes. They exist in nature, and they are able to use carbon dioxide and carbonates. Heterotrophs are bacteria, and as carbon source, carbon-containing compounds are used. Hydrocarbons, polyhydric alcohols, amino acids, and organic acids. They are named heterotrophs. And at the same time, heterotrophic bacteria are divided into saprophytes and parasites. Saprophytic bacteria, metatrophs, grow on dead organic residues. They exist in nature, saprophytic bacteria. They are able to get substances from the dead organic particles. Parasitic bacteria, paratrophs, feed on organic compounds of living organisms. So they are adapted to the life inside the animal's body or human body. And so the most of pathogenic bacteria, of course, are parasites of human body or animals, and they belong to paratrophs. The second, breath of bacteria. All living cells need oxygen, and bacteria are living cells, prokaryotic cells. Bacteria are able to get oxygen indirectly from hydrogen peroxide or CO2 molecules indirectly, and some bacteria are able to get O2 directly from the molecular oxygen, O2, free oxygen, air oxygen, molecular. And bacteria can exist under oxygen conditions in the presence of bacterial enzymes that neutralize toxic oxygen compounds. So, if bacteria exist in the presence of molecular O2, they are due to metabolism, they will develop substances, for example, hydrogen peroxide, H2O2, and that is toxic substance for a bacterial cell. And if bacteria has enzymes for destruction of this toxic substance, hydrogen peroxide, bacteria are able to survive. Most of pathogenic bacteria have got such enzymes. They have many enzymes, and so they are able to exist in the presence of molecular oxygen. It's important to know about pathogenesis of infections. Most of bacteria are able to survive on the mucosa membrane where molecular oxygen is, and they don't die. And it's important to know for my bacteriological method for diagnosis, microbiological diagnosis of bacterial infections. Most of bacteria are cultivated in presence of O2 because they are able to multiply, to grow up in presence of O2. But some bacteria are not able to survive in presence of O2 because they have no all enzymes. Those are necessarily for destruction of toxic products. And memorize, it's very important, in relation to O2, molecular O2, bacteria are divided into 5 groups. Group number 1, obligate. Obligate means strict. Obligate aerobes need molecular oxygen, O2, and they are able to survive. And so they are cultivated in ordinary test tubes closed by cotton stoppers, and they are cultivated in petri dishes. Those are not tightly closed because they need O2. For example, Pseudomonas aeruginosa, bacteria of Pseudomonas genus, they need O2, and they very often cause hospital outbreaks. They are very resistant to environmental factors, and they are able to cause inflammation with spasms in any organs. Number 2, bacteria named obligate strict anaerobes do not grow in the presence of O2. For example, are Clostridia genus bacteria, Clostridium, causative agents of tetanus and botulism. Strict anaerobic bacteria have no all enzymes, so they are not able to destroy toxic products. Those are developed in reaction with O2. And finally, they die of toxic products of own metabolism. And we should think about pathogenesis of tetanus and botulism. These bacteria, if they multiply in human's body, they need conditions without O2. Tetanus is wound infection, and in necrotic tissues, these bacteria are able to multiply. So for non-specific prevention of tetanus, surgery should be done, proceeded to remove necrotic tissues if it's possible to avoid multiplication of strict anaerobic bacteria. And causative vegetal botulism is able to multiply in the wound because of they develop spores, you know, from laboratory classes. Clostridia bacteria, those are able to develop spores. And contamination by these bacteria started from the spores entering. So if spores are in anaerobic conditions and they germinated, and bacteria are able to multiply. But if there are aerobic conditions, bacteria will die because of they are obligate anaerobic bacteria. Botulism very often like food poisoning. Again, bacteria are able to multiply just in absence of O2 in anaerobic conditions, maybe canned food, maybe sausage, and they are able to cause wound infections, let's say. Then the third group of bacteria is named, bacteria named facultative anaerobes. For example, Staphylococcus genus bacteria, Escherichia genus, they grow in both conditions. Grow and multiply in the presence and in the absence of O2. Why? Because of, and most of pathogenic bacteria, causative agents of infectious disease of human beings, are facultative anaerobic bacteria because of they have a complete set of enzymes and they are able to destroy toxic products. Those are developed due to reaction with O2. So they are able to survive in presence of O2 and in the absence of O2. And in laboratory, in bacteriological laboratory, they are cultivated in presence of O2 because it is easier. They are cultivated in ordinary petri dishes where oxygen is and in test tubes closed by just cotton stoppers. They are able to survive because it is difficult to create anaerobic conditions. Anaerobic conditions are developed just for cultivation, isolation of this Clostridia, causative agent of tetanus, causative agent of botulism, and the third one. And then you will study how it is possible to create anaerobic conditions. Then there are bacteria that are named air-tolerant bacteria. They grow in the presence of O2 for a few hours, but do not use it. Then example is the causative agent of gas gangrene. These bacteria belong to Clostridium genus. They are Clostridias, but they are able to survive a few hours in presence of O2, and it is used again in diagnosis of gas gangrene. These bacteria are cultivated in the test tubes closed by just cotton stopper. But if we are going to isolate pure culture, anyway, we should create anaerobic conditions. They are not able to survive for a few days. And so, because they belong to Clostridium genus. So Clostridium genus bacteria are not able to survive in presence of O2. Microaerophilic bacteria need oxygen, but grow better in presence of CO2. Microaerophilic bacteria need small amount of O2, and they grow slowly if we will create conditions with CO2 by special commercial gas pack, special packs with chemicals inside, and it is possible to create conditions of CO2. They grow faster. Bacteria of Campylobacter genus and Helicobacter genus, you did draw a picture of them, they are spiral bacteria, and bacteria of Brucella genus, they are microaerophilic bacteria. So there are 5 bacteria divided on 5 groups in accordance with oxygen requirement. And of course, it is necessary information for cultivation of these bacteria in bacteriological laboratory. Then the third that we should know, growth and reproduction of bacteria. The growth of bacteria is an increase in cell mass and an increase in the number of cells as a result of division. So the growth of bacteria, just cell is increased, and reproduction of bacteria means increase the number of cells. They are able to multiply by binary division in human's body, in vivo, and then in laboratory, in artificial conditions, in vitro. So we should know what factors they need for better division, for better growth. When we are going to isolate the presumptive causative agent, suspected causative agents, in accordance with diagnosis, we should know what factors affecting the growth of bacteria, because we should create favorable conditions for them, for their growth. So we should know what the best conditions they are able to grow up. Number one, we should realize cultivation method. Maybe artificial media, many bacteria grow up on artificial media, different content, maybe simple media, maybe enriched media, but they are able. Some of them need cell culture. If bacteria, those are obligate intercellular parasites, they are not able to grow up on artificial media, and they are Rickettsia, bacteria of Rickettsia family, chlamydia, bacteria of chlamydia family, and causative agent of leprosy. These bacteria are not able to multiply on artificial media, so they are cultivated like viruses. They are bacteria, but they need the host cell for multiplication. So they are cultivated either on cell culture, maybe by using of laboratory animals, and maybe chicken embryo. Chicken embryo means an embryonated, hence an egg. An embryonated, hence an egg, is named in short chicken embryo. So they are cultivated like viruses. Special methods are used for them. Then we should realize what temperature is favorite for these bacteria growing up. And temperature conditions distinguish three groups of bacteria. Mesophilic bacteria from 20 till 40 degrees by Celsius, and most pathogenic for humans, need the human's body temperature. Of course, they are adapted to humans being temperature, and so the temperature as usual is used 37 degrees by Celsius. This is favorable conditions for them. Some bacteria named thermophilic bacteria from 50 till 60 degrees, and these bacteria exist in nature. They participated in destruction of organic substances. They play a very important role on the earth. Among pathogenic bacteria, just Pseudomonas genus bacteria are able to exist at temperature a little bit more than 40 degrees by Celsius, and some thermophilic strains of Escherichia, just a few of them, are able to exist at temperature 44 degrees by Celsius, for example. And psychrophilic bacteria, they prefer low temperature from 0 till 10 degrees by Celsius, and the causative till 10 till 20 degrees by Celsius. And again, most of them exist in the nature. And among pathogenic bacteria, causative bacteria of Yersinia genus are psychrophilic bacteria, causative agent of plague, causative agent of plague, causative agent of pseudotuberculosis, intestinal Yersiniosis. You will study them. They belong to one genus Yersinia, and the favorable temperature for them is from 0 till 25. Exactly 25 temperature. 28 is used for cultivation of causative agent of plague, Yersinia pestis. So they are like an exception. Then when we are going to create medium for pathogenic bacteria isolation, we should create the 13 pH. As usual, most of bacteria, they need neutral pH from 7.2 till 7.4. But there are an exception for the isolation of Vibrio cholera, causative agent of cholera. Alkaline medias are used, and pH should be 7.8 at 0 needed. So really, these bacteria are able to grow up on very simple media, such as 1% peptone water. 1% peptone is enough for these bacteria, but pH should be alkaline. They are able to grow up on a simple basic medium nutrient agar without any other substances, such as blood, sugar, they don't need, but pH should be alkaline. So alkaline media are special for isolation of causative agent of cholera. Then, and when bacteria are inoculated on media or cell culture, they should be cultivated, incubated in special equipment named thermostat. Thermostat is used for cultivation of bacteria, and it works automatically, clock around day and night, like refrigerator. And it is possible to set the proper temperature, and this equipment, this thermostat, will keep the proper temperature constantly. Very often the temperature is 37 degrees by Celsius, but it depends, for causative agent of Yersinia, for isolation of Leptospira, the temperature should be 28 degrees by Celsius. So we should know concentration of sodium chloride. For most of bacteria, concentration of sodium chloride, as for people, for human beings, 0.9%. Such solution is named normal salt solution, and it is used for human beings, and the same concentration is used for bacteria. But again, there is an exception, and always you should memorize an exception. Bacteria of Staphylococcus genus, Staphylococci, they are able to grow up in presence of 9% of sodium chloride, so 10 times more. And Staphylococci very often cause inflammation with pus, and when we are going to isolate Staphylococci, of course, selective medium should be used, salt agar with 9% sodium chloride concentration. Staphylococci are able to grow up, other bacteria are not able to multiply too much sodium chloride. Then, after we will inoculate bacteria, and then we will place media in thermostat, then we are waiting for visible growth. Before inoculation, media, liquid media, solid media should be sterile, and they are clear, not possible to see any signs of growth. But after incubation at thermostat, we should check. We are looking for visible growth. So it is necessary to know what time is required for the appearance of visible growth on nutrient media. For most of bacteria, it takes 18-24 hours. In average, some bacteria, again an exception, grow up in 6-8 hours. For example, causative agents of cholera and diphtheria. Other bacteria grow after a few weeks. For example, Brucella, they grow up during 4 weeks, and in presence of CO2, we will see visible growth in 2 weeks. Leptospira needs a few weeks till 2 months sometimes. And some bacteria are able, they multiply very, very slowly. They divide very slowly. So visible growth appear after 1 month, maybe 2 months, and even 3 months. And you should memorize what are they. They are causative agents of tuberculosis, bacteria of Mycobacterium genus, Mycobacteria. So in case of tuberculosis, the positive answer from bacteriological laboratory may be sent in 1 month, in 2 months, but we have to wait 3 months and for negative answers. If there are no any signs of growth, in 3 months, a bacteriologist will write negative answer, like a causative agent of tuberculosis was not isolated. They divide very slowly, these bacteria. So how it is looking, the visible growth of bacteria. For cultivation of bacteria, liquid media are used and solid nutrient media are used. Liquid media should be clear in test tube or maybe in some bottles. For blood inoculation, for example, the bottle should be used. And before inoculation, liquid medium is absolutely clear. And after inoculation, we are looking for some signs of visible signs of growth of bacteria. So different if bacteria multiply in liquid media, they are able to change medium differently. So, for example, bacteria can form the following forms of growth. Uniform turbidity and sediment in the bottom. Uniform turbidity means completely unclear medium. It was clear and now it is unclear, and maybe some particles on the bottom. So most pathogenic bacteria multiply with uniform turbidity, unclear medium. Some bacteria don't multiply, are able to multiply in liquid media, but they don't cause any visible changes. Leptospira, they are cultivated in the serum or blood of rabbits, and they multiply, but they move actively. Leptospira spiral bacteria, you know them, you did draw a picture of them. They move actively, so they are not concentrated on some place. And this medium, serum or blood, still absolutely clear. So we should do microscopy every week to realize do they multiply or not. Then some bacteria develop a film on the surface of the medium, a layer, like causative agent of cholera, the causative agent of diphtheria, causative agents of tuberculosis. On liquid medium, they develop a layer on the surface of medium. And then some bacteria develop the layer on the wall and the bottom of test tube, such as streptococcus. The medium still clear, but it is possible to see the visible layer on the wall. And some bacteria grows in the form of lumps of the cotton wool, like causative agent of anthrax. The liquid medium still clear, but it is possible to notice some particles in this liquid medium. But very often, the primary inoculation of material is done on solid medium. Solid media, it is possible to get solid medium if we will add agar-agar. And agar-agar is polysaccharide of the seagrass, and this agar-agar gives the is able to create solid medium. And solid medium is placed in petri dishes, and inoculation is done on the surface of this solid nutrient media. Bacteria form macroscopic accumulations named colonies on the surface of the medium. Each colony, sometimes the species is inoculated inside the medium. Anyway, bacteria are able to develop colonies. Each colony is formed by bacteria of one species as a result of the reproduction of one or more bacterial cells. Or there is photo of petri dishes with solid medium. In this picture, in this photo, this is blood agar, because some bacteria are not able to grow up on basic medium, just nutrient agar. They need some substances such as blood. Some bacteria need blood agar, so it is red or blood. Some bacteria need sugar agar. Some bacteria need serum of blood. Some bacteria need a lot of substances for growing up, need very enriched medium. So every spectacle is formed by a bacteria of one species, and they are named colonies. Colonies of different species of bacteria differ in size, shape, surface, consistency, nature of edges, colors, and so on. They are different, and some of them are smooth, some of them have irregular shapes, some of them have rough shapes, some of them have smooth surface. So in microbiology, they are divided on two types. There are S and R type colonies. So S type colonies from the word smooth. S type colony are round, smooth, shiny, convex, with smooth edge and moist. R-type colony from the word rough. So the type of colony is larger in size, then they're flat, the surface is rough, neat, and irregular in shape with straighted edges. So, for example, this is photo of S-type Escherichia colonies. The S-type colonies, you see, what are they? Smooth surface, elevated colonies, regular surrounded edges, so S-type colonies. Most of pathogenic bacteria develop exactly S-type colonies, most of them. Just 5 causative agents are able to develop R-type colony. For example, there is a photo of R-type colony of the causative agent of tuberculosis, looking like some berries, and causative agent of anthrax develop R-type colonies, causative agent of diphtheria, R-type colonies, daisy flowers, causative agent of mycoplasma, fried egg colonies, and causative agent of plague. So 5, you should memorize 5 causative agents because they develop R-type colony. It is important in diagnosis because of after inoculation of specimen, we should know what colonies are suspicious, what colonies are we looking for. Very often we are looking for S-type colonies, and in case of these 5 infections, we are looking for R-type colonies. So memorize 5 causative agents. Then physiology of bacteria study enzymes. Enzymes of bacteria. Enzymes are specific protein catalyst present in all living cells. They are living cells, they need enzymes for metabolism, of course, and for each transformation of one compound into another, a particular enzyme is responsible. And detection of enzymes is important for identification of bacteria. Identification means detection of species name. What species do these bacteria belong to? Really, we are going to know, do they belong to the presumptive species? For identification, many properties are necessary to study, such as, firstly, type of colonies, cultural properties, then morphology of bacteria, we should do smear of these bacteria, then sometimes it is necessary to detect enzymes. Not always, but sometimes it's necessarily like bacteria. Those belong to Enterobacteriaceae family, Enterobacteria, genera Escherichia, Salmonella, Shigella, Klebsiella, Proteus. They are not differ by morphology and cultural properties, and it is necessary to study enzymes, biochemical properties, and antigenic structure for identification. And then pigments. Some bacteria are able to secrete pigments, dyes, by own metabolism. So colonies of such bacteria on solid medium are painted, always have color due to pigments production. And for example, bacteria of Staphylococcus genus are able to produce pigments. Bacteria of those belong to Pseudomonas aeruginosa species, Pseudomonas, are able to produce pigments. So doesn't matter what media do they grow, what content of medium, colonies of such bacteria always have color. It depends on the color of pigment. For example, Staphylococci are able to produce pigments white color, golden color, yellow color, and bacteria, Pseudomonas genus bacteria, they produce pigments of different colors, mainly greenish bluish, maybe red color, maybe brown color. Anyway, so in case causative agents of diphtheria are able to produce on slightly brown pigment. Causative agent of tuberculosis, again, slightly brown. So it's important for identification of bacteria. For example, pigmented S-type colonies of Staphylococci, and they grow up on salt agar. I told you, salt agar is used like selective medium, and they have always color, yellow color, white color. They are pigmented, and there is a photo of colonies with pigments. They are normal microbial flora of air, by the way, and very often bacteria that are able to circulate in the air develop colonies with the color. And you see different types of colony. There is R-type colony, for example. Now we will make a break, 5 minutes break, and then in 5 minutes we will continue the lecture by using of the same link. Do you hear me? You should join the lecture in 5 minutes by using of the same link.
Okay,
Do you
doctor.
hear me?
Yes, doctor.
Yes, okay.
Okay, so in
Yes,
5 minutes,
okay.
the lecture will be continued. So 8:45, join the lecture again. 8:45.